Hello and welcome to this PIR Live Event webinar, brought to you in part by Partners in Research Canada. My name is Ben Hobbs, and I will be your host today. If you're joining us live, remember that you can ask questions at any point by clicking Q&A, which is located in the bottom center of your Zoom pop-up window. I will relay these questions to our guests during the Q&A portion of the webinar. If you see a question in there someone has asked that you would also like the question to, you can prioritize it by clicking that thumbs up icon just below their question. So without further ado, I would like to introduce our guest today, Dr. Michelle D'Souza, postdoctoral fellow in the Center for Biodiversity Genomics at the University of Guelph. Thanks for joining us, Dr. D'Souza. I'll let you take it from here.
Thank you very much, and hi to everyone who's tuned in today. I'm very, very excited to let you all know a little bit about what I have been working on the past few years. So I'm just gonna share my presentation for you, and hopefully it all goes well. Okay, am I good to go?
That's perfect, thank you.
Wonderful, all right. So I wanna start by asking the question, where have all the insects gone? And I want you to think about this because this is something a lot of researchers have been thinking very hard about lately. Because we've had a lot of studies that have been indicating that there's a really dramatic decline in insects in the world right now. So one of the most recent studies that came out talks about almost a 40% decline in insect species. So this graph here with the different colors is just indicating the level of danger that we see some of these different insects groups. And why does this matter? Well, it's really important because insects are very, very vital to what we do. They are very key in a lot of pollination processes, making plants do what they do. And this is then linked to the amount of food that's available for us. And directly, insects themselves are actually a very large food source for other animals such as birds and frogs. They're also very important for carrying diseases such as malaria. And so they really are a very, very important part of our world. But there's a couple of things we really need to consider and understand if we need to know whether this decline in insects is truly worldwide. And the first thing is that we really don't know very much about the variety of species or the biodiversity of these insects. Our knowledge is very incomplete. So it's estimated that there are about 10 million species on our planet, and we really only know about 20% of them. Insects making up a very large portion of them. To give you an example, we have more ant species than we have bird species. They're very, very diverse. So the other big problem really is that when we're looking at these declines, what we know is very concentrated in certain parts of the world, specifically Europe and North America. So we really don't have a lot of data to truly understand whether this is a worldwide global issue. And I'm going to just highlight the continent of Africa here because that's where the rest of my presentation will talk. There's a lot more data that we need to collect. And so that brings me to the Kruger Malaise Program, what I've been working on for the past year. And this is a program that's really bringing park rangers, staff, and scientists together to better understand insects in South Africa and hopefully in the larger African continent. So once again, my name is Michelle D'Souza. I'm a PhD student. Sorry, I'm not a PhD student. I'm a postdoctoral fellow, recently graduated, working here now at the Center for Biodiversity Genomics at the University of Guelph. And what I'd like to start talking about today first is how we can start to address some of these challenges. And then I'd like to give you a few more details about the Kruger Malaise Program, and then leave you with a couple of final thoughts. All right, so as I said, there's two kind of big challenges that we're facing. One is that we only really know about a very small portion of insects. And what we do know is very isolated to particular regions. So how do we get better at understanding and identifying a lot of these other species that we don't know about? So one of the things that was developed here at the University of Guelph from Paul Bear was the ability to use DNA barcodes, which are short sequences of DNA that can be used to identify species. So all living things have DNA, and DNA barcodes really have shown that we only need to read a very small portion of that DNA in order to be able to identify a species. And so the process is actually very, very simple. So you start by first extracting that DNA, You then isolate and make copies of that DNA barcode. And then you read that DNA barcode through a process called sequencing. You then match that DNA barcode to a reference library, and there you have it. You know what species you're working with. And why use DNA barcodes? Well, they're a lot faster and cheaper than a lot of the other methods out there to identify species. And importantly, it's very digital, and that's really important for automation, which means we can take advantage of technology and DNA barcoding DNA technologies in order to do things for a lot cheaper, faster, and more effective. But our other bigger challenge is, how do we get to all of these areas where we know nothing about? And here, international collaborations are a very, very important part. So the National Barcode of Life was established in 2010, and this is an alliance between 30 nations that are working to address some of these deficiencies in our knowledge on biodiversity. And so far, the International Barcode of Life has been sampling all throughout the world, gathering data to better understand biodiversity on our planet. And for the rest of the talk, I want to focus in on one particular country, South Africa, which is where we have been doing the Kruger Malaise Program. Okay, so let's get into it. So the Kruger Malaise Program, why South Africa? Well, there is a large shortage of data on African insects. And more importantly, we have a really good collaborator there in South Africa, the African Center for DNA Barcoding. And this institute represents the largest DNA barcoding facility on the continent. So just a very, very good place to start this type of work. Why Kruger National Park? It's the largest of 19 national parks in South Africa and the home for the scientific services of the South African National Parks, our other collaborator. And the Kruger Malaise Program has the word Malaise in it, and that's because we use Malaise traps to collect insects. And why do we use them? Because they are very simple and allow us to gather data that is comparable across different sites. So how a Malaise trap works, it's basically a tent, if you want to think of it, and insects fly through an area, they hit the wall of that tent, and they get funneled into a collection bottle, which can then be collected. And why do the Kruger Malaise Program? Because we're interested in examining how the number and different types of species change over time in a very large national park, and hopefully use this as a model to then go out and sample other national parks, not only in South Africa, but across the world. Okay. So let us find out a little bit more. Okay, so the Kruger Malaise Program officially began in May 2018 when I went to Johannesburg, met up with colleagues from the African Center for DNA Barcoding, and traveled to Kruger National Park. So we got into our soccer mom van and we went all the way from the south of the park to the north and deployed or put down 26 Malaise traps. And we did this within rangers' properties because of the possibility of wild animals getting to the traps had we left them out in the wild. And one of the best parts about this project is all of the different people that I got to talk to. So there are rangers working in each of these different areas, working to protect the wildlife within the park. So these are animals like your rhinos, your lions, your elephants, and all of these people spend a lot of time protecting that diversity, and it is not often that they get to participate in a project like this. So they were all very, very excited to participate. And we really couldn't have done it without them because it was the rangers who were actually collecting the samples for us. And they go out to these traps every week for a full year, and they've been collecting these insects and giving us this mass amounts of information about what's happening in the park. So what happens after they collect it? So we go from Kruger, and our colleagues at the African Center for DNA Barcoding drive them over to their lab in Johannesburg. And from there, they prepare it for shipment. And they send it to us here at Guelph, Ontario, to the Center for Biodiversity Genomics, where we sequence all of these samples and provide some results. So we've had 3 shipments so far, one in August, one in December, and one in March. And that's accumulated to about 925 insect samples that have been shipped to us so far. So 925 samples that the rangers have collected, that the African Center for DNA Barcoding have transported from Kruger and shipped to us here at Guelph. So we've sampled a portion of those so far from the first 2 shipments. And this is kind of what that sampling process looks like. We take those insects, we put them into a petri dish, which is what you see in the middle here, and look at them under a microscope. And then we isolate every single individual. When they're larger, we pin them. And we only take a very small portion of the animal in order to get its DNA. So we actually leave the animal intact so that we can go back to it and look at other types of traits if we wanted to. And once again, like I said, the process is pretty simple. We go through it, find a barcode, and then we identify it. And so far, we have processed 180,000 insects. So what have we found so far? There's a lot of variation in the types of insects. So this is data just from the shipment from August, which has been finished sequencing. And this is a graph here of the number of species that we've found so far. And so the length of the bars kind of give you an indication of how many species we have. And out of the 64,000 that we sequenced in our first shipment, we have about 6,000 species that we've found across the park. And what's really interesting is that about 70% of those species are only found in one trap. So you can see that there's a lot of unique insects across this park. And of the species that are shared, you can see that there is an incredible network of connections. So this is just a graph to illustrate how intricate that connection is. So each of those letters represent one of those traps, and all of those lines are the connections of shared species. So there's a really big network of interactions that are happening here that we have the opportunity to delve into. And I can't go into too much detail, but to just give you a quick overview, here's just a network to show you how those traps are related to one another. And you can see that here, they kind of segregate into these 2 colors, green and yellow. And those 2 colors are the big ecoregions in the area. So there's the data aspect of it, but the other really exciting part was getting to talk to the rangers and seeing how they interact with the project and what they think of it. So I spoke to Greg Bond, one of the rangers in the South, and he said that, this project has been really interesting because it's made him way more aware of the insects around him than he's ever been before. Palile from the North, one of our section rangers, said that it's not often that scientists actually come into the park and get that involved with the rangers. And so she was really excited to learn about the process of science. So she actually was applying to do her PhD as well. But most alarming for me was when I got to talk to Robin Peterson from Kruger, who's a freshwater ecologist. And while the data is lacking, he did tell me that he's noticing that there are declines, or he is noticing that there are differences in the insect populations within Kruger, saying that he actually sees less and he hears less than 10 years ago. So that is cause for concern. And hopefully the Kruger Malaise data that we collect will provide some and shed some light on what he hears and sees. And so just to end off on some final thoughts for you guys. Insects are a very important part of our world. And there is a widespread decline. Widespread declines of insects are a really big concern. So we really need to better understand and monitor species. And international collaborations are very important for this work, but you're needed as well. The more you understand and the more you care about insects, the more we can tackle some of these issues. And with that, I want to say thank you, and I very much look forward to the questions you have for me.
Well, thank you, Dr. D'Souza. Just before we move into the Q&A today, I'd like to remind those classrooms who are joining us live to direct their questions to the Q&A, which you can find in the bottom center of your Zoom window. So our first question today comes from Mrs. Moore's students in Kingston, Ontario, and they're wondering, why is it that insects have more diversity than plants or animals?
So that's a very interesting question. And, it might just be because they are a lot smaller and they fill more niches. And niches are kind of these spaces that that kind of create a role for an organism. So insects just have versatility. They are hardier of an organism. They have a hard exoskeleton is what we call the outside. So it's just likely because they fill a lot more of these roles. They have a lot of diversity to attack a lot of these issues that we see in the environment.
Earlier in your presentation, you mentioned how DNA barcoding is a great alternative because it's cheaper. What is it cheaper? What are the alternatives, and why? What makes it cheaper?
Yeah, so traditionally how we've been studying species is by actually looking at them under a microscope and describing morphological characteristics, so differences in the color, the size, the shape. And this takes a lot of time, but it also takes a lot of specialization. So the people who are actually trained to do this type of work are becoming rarer and rarer. So by using DNA, it's a lot faster, and it's inexpensive and takes a lot less time than all of these other ways of describing species.
The students joining us from Aurelia are wondering what might be some of the reasons why insects are in the decline in Kruger.
Yeah, so we really don't have a lot of data to really know. There's a lot of theories out there. One of the bigger ones currently is agricultural pressures. So there's a lot of land transformation that's happening. We're a growing population and we need a lot of food to survive. And along with agriculture comes a lot of pesticide use. And so there's a lot of theories out there that this might actually be affecting insect populations. Climate change is another big one. Insects can be quite sensitive to changes in temperature, for example. But really, as I kind of hope I've alluded to, there's a real gap in the data. We really don't know.
So our next question today comes from Hope, and she is wondering, are the animals alive when the DNA is taken for the DNA barcoding?
No. So the animals are not alive when the DNA barcodes are taken. So when they go into our trap and they're collected, they actually die very quickly. So they stay and are preserved in ethanol within our traps. So they're not alive for very long once they're collected.
Zach from Mrs. Bonacard's class is wondering, what do your results tell us about the health of our ecosystem?
So far, I haven't had the chance to go into the results too much, but when we look at, like I said, all of these other studies, it does seem to indicate that we might have an issue on our hands. And that's the really important part of this type of research. We need to better understand what might be causing these differences. And right now, we honestly don't even really understand what the baseline level of a lot of these insect populations are. So this work is really crucial for a place like Kruger to just get an understanding of what insects are doing, so that if something bad is happening, let's say in the future, or has been happening right now, we'll actually be able to better assess those changes.
This decline that we might be seeing in South Africa, is this consistent with insect populations around the world, or do you know if this is just isolated to specifically South Africa?
Yeah, so actually we haven't seen these declines in South Africa yet. And that's one of the really interesting parts of this project. So it's going into an area which for the most part should be a lot less affected by human impact than places like Europe, where these declines have been measured and are being detected. South Africa has a lot younger of an agricultural industry as well, and we are sampling within a national park. So I would say through this project, if we do see some sort of trends of decline, I think it will be a really big indicator of this being a very big problem, because, you know, if there are places or reservoirs in the world right now where this isn't happening, I would imagine that they would be in these natural intact systems like national parks. But yeah, we don't actually have that, we don't have that data yet. What we do know is that the rangers and the staff who have been working in Kruger for so long are hearing differences, are seeing differences. And people all over the world are saying that as well. You know, there's this analogy right now that, I mean, I would love you guys to ask your parents if they feel this way. A lot of people go for drives now and they say that they don't have as many insects hitting their windshields as they used to a few years ago. So this is stuff that people are noticing, but we need the data to actually back up all of these things.
So would it be possible, number one, to barcode all the species, at least that we know of on Earth? And do we have any sort of timeline for how long that would take?
That's a great question. And yes, so the International Barcode of Life has the very, very ambitious goal of actually barcoding all multicellular life on our planet, so animals, plants, and fungi. And we want to do this by 2045. And once that's in place, there really is a very big potential to ask a lot of great questions. For example, one of the big things that we can and will be able to do if we do have this complete reference library is be able to better monitor, for example, ports of entry. So we'll know when invasive species are coming to a country because we'll be able to detect them because we have them in our reference library. So there's a lot of different applications that we can start to kind of, and a lot of challenges we can start to tackle. Hopefully by 2045 with the support of the international community. So yeah, this is something that we do want to do, and we do want to make it a reality. It isn't science fiction for us.
Our next question is from Matthew, and he's wondering, if you take a sample from a specimen with a genetic mutation, would it affect the identification of that species?
Awesome question. Yeah, so genetic mutations are evolution. Yeah, and that's exactly why DNA barcoding works. So we've picked a very specific region, and you have to consider all of these different things in order to find the right one that will be just right enough and doesn't change fast enough for us to be able to differentiate species. And that's exactly what the DNA barcode has been shown to do within certain groups of organisms. Yeah.
Alyssa is wondering, how do you plan to find or discover the other 20% of insects you have not yet identified?
Yeah, so one thing I think that is really important and more people need to do is help us do this. So we had a couple of years ago a program called the School Malaise Program, for example, where we actually went out into schools and set malaise traps in each of schools around Ontario. Everyone can get involved. That's the really exciting thing about DNA barcoding. You don't have to be a top level scientist for this to work, for this to make sense. Everyone can help collect all around the world. And that's kind of what we're hoping for. That's what the International Barcode of Life is hoping to do, promote this type of work and get more people involved, because this cannot be something that just a few of us do. Everyone needs to get involved.
Is there anything that we can do as average citizens to help keep these insect populations from declining in our own communities?
Yeah, I think about this a lot, and there's a lot of kind of tangible things you can do. But for me, I personally think one of the biggest things is that we don't have an awareness of insects as much. I hear people talk about insects more negatively than I do positively, and I think that's kind of the biggest step. It's really just thinking about how you interact with insects, thinking about how insects interact with your life, and kind of reevaluating some of the things you think about them. I think that is probably the biggest step, because at the root of all of this, it's a connection that we have with nature, and kind of building that connection is going to be the basis for everything else that we do. So I just encourage everyone to just learn a little bit more about the animals around you and care a little bit more. And then, from there, reach out to your local organizations in your communities and find out how you can get involved based on how you feed off that connection.
So Hannah is wondering, what other information can the barcodes tell us besides what the species is?
So there are a couple of other things that it can tell us, but the barcode really doesn't try to get too much into, evolution and that kind of stuff. What the barcodes really are best at are identifying species. But because of the barcode process, and by this I mean we don't, we keep every organism that we get a barcode from. So what this allows us to do then is go in and look at other genes. So other genes are sometimes better at telling us a better story of what this organism is. So while we collect all of this information and we curate it, kind of like you have those IDs in a library to find the book that you want, that's what a barcode is really doing. It's organizing this library so that then when you want to know a little bit more about one particular animal, you know exactly where to go to find it, and then you can open the book and read more as you want to.
So we've got a couple of minutes left today, but before we end, I was hoping to have you speak a little bit about your career path, how you ended up where you are today, and maybe some advice for students who are watching, considering something similar for their own futures.
Yeah. So, you know, I spoke about that idea of connection, and for me, that's really where it started. So I grew up in India, and I was running around in the forest in India growing up, and for me, that was my playground, you know, and I developed a connection with animals then. And I kind of went into a science career because I also found that I was really good at understanding science questions. But I really could have taken any other path and still stayed to that connection. I really like to cook, for example. I could have become a chef and talked about sustainable food. But I decided to go down a science route, and I ended up getting a PhD because, once again, it gave me the opportunity to go back into those environments, into those forests that I loved so much, and connect with nature. And by using science, I feel like I am better equipped to actually then go back and protect it. And so I went to the University of Guelph, I got my PhD, and I was lucky enough to start with a postdoc here. And since then, I have just been very, very much involved with telling the story of science and getting more people involved and strengthening that connection that we have with nature.
Well, that's all the time we have for today, but thank you, Dr. De Souza, for taking the time to showcase this great international collaboration and for answering all of our questions.
Thank you very much, everyone. I hope you learned something today.
So next time on PIR Live Event, Tim Thurman and Madeline Stange from the Barrett Lab at McGill University are joining us to talk about how animals adapt to changes in their habitat and environment. More information about these webinars and other PIR educational programs are available at pirweb.org. Thanks for tuning in and have a wonderful day.